Let's now talk briefly about lipids and the important role that they play within the membranes of living cells. So some of the concepts we'll focus on listed on the screen for you at this point, but let's begin with just making the point that it doesn't matter what type of lipid you're talking about. One feature that all lipids share in common is that they are all hydrophobic, meaning they just don't mix well with water. So again, that's a feature common to all lipids. We can identify at least three major types of lipids, namely fats, steroids, and also phospholipids. So let's now talk briefly about the structure and function of each of these types of lipids. Beginning with fats. Fats, as you might guess, they have lots of calories, and they of course play a big role in storing energy. Right? So this of course is something that many people are aware of in terms of their nutrition, their diet, right? But of course, fats have lots of energy. Structurally, what are fat molecules like? Well, we can describe fats as consisting of glycerol plus 3 fatty acids. So what exactly does this refer to? Well, if you take a look at this image here, illustrating the basic structure of a fat molecule, glycerol is this 3-carbon backbone, highlighted in gray, but then from each of those 3 carbons, we see this long chain of carbons, illustrated by this black line here. These are all fatty acids. So again, fat molecules, basically glycerol, each carbon having a fatty acid extending from it. A little different look at fat molecules, again, 3-carbon backbone. Each one has a long chain of hydrocarbons. So these are the fatty acids. We can talk about saturated fats, like we see here, meaning that each carbon is saturated with as many hydrogens as it can possibly hold, or unsaturated fats, meaning that, notice some of the carbons are double bonded to other carbons and therefore are not saturated with as many hydrogens as they can possibly hold. Another type of lipid that we'll identify are steroids. So steroids actually are a type of lipid. Cholesterol is actually a type of steroid. Various hormones, they too are steroid in nature. So we're not going to dwell on steroids at this point in terms of how they work or what they do. I just want to briefly point out the structure of all steroids. They all share this very characteristic 4-ring structure in common. So more about the function and the significance of steroids a bit later on. The third major type of lipid that we identify are phospholipids. It turns out that phospholipids, because of their structure, they are the major component of cell membranes. So what makes phospholipids so important or so adept at being part of cell membranes? Well, it's hard to illustrate in this particular figure, but what we're looking at is a phospholipid. So here's our 3-carbon glycerol in dark gray here. 2 of those carbons have fatty acids linked to them. So we see 2 fatty acids there. The 3rd carbon, however, actually is linked to a phosphate group. So what are the implications? Well, it turns out that the fatty acids, they are indeed hydrophobic, so they don't mix well with water, but the phosphate group is hydrophilic. It tends to be attracted to, or it mixes well with water. So here we have these single molecules with both hydrophobic and hydrophilic regions. What are the implications? Well, when you take a bunch of phospholipids, add them to an aqueous environment, so add them to water, it turns out that the hydrophobic fatty acid chains orient themselves away from the water, whereas the hydrophilic phosphate groups orient themselves toward the water. So what we get is this bilayer of phospholipids, which essentially represents a cell membrane. So, as you can imagine, cells have lots of things that have to move either into the cells or out of the cells across cell membranes. So let's briefly talk about basic concepts of movement of solutes and water across cell membranes. So terms you've heard of before, things like diffusion. Diffusion is simply the movement of molecules, typically from a region of high concentration to a region of lower concentration. It just relies on ambient energy. It doesn't require any extra energy to take place. Osmosis is simply the movement of water across a membrane. So you might think of it as diffusion of water across a membrane. Okay, so let's explore these concepts in a bit more detail. So think about osmosis. Think about movement of water across a cell membrane, and we can talk about the concentration of solutes. In other words, the tonicity of a situation in question. So, for example, we can identify three situations. First of all, we often have a situation where the concentration of solutes inside a cell is the same as the concentration of solutes outside the cell. We say this is an isotonic situation, right? Notice that we always have to have two reference points in question, at least two, perhaps more. But in this case, we're talking about inside a cell versus its surrounding environment. So the concentration of solutes, if they're the same in both places, we say that's an isotonic situation. In contrast, if we have a situation, again, if we're comparing two reference points, whatever region has a higher concentration of solutes, we say that region is hypertonic, whereas the other region would be hypotonic. So a hypertonic region has a high concentration of solutes, whereas the hypotonic region has a lower concentration of solutes. So the extreme version of hypotonic would be pure distilled water. So don't let the terminology confuse you. These are profoundly important concepts. Really, organisms have to deal with tonicity and diffusion osmosis on a regular basis because it really impacts a whole host of physiological conditions. So let's illustrate these basic concepts as seen in the image on the screen. So first of all, focus on the far right. So we see a cell, notice the phospholipid bilayer membrane there, and some surrounding solution. And let's say again on the far right, the concentration of solutes, illustrated by the green spheres, is pretty much the same inside the cell as well as in the surrounding environment. So this is an isotonic situation. What we're focusing on in this image is the movement of water. Not so much the movement of the solutes, but the movement of water. I want to point out that in this isotonic situation, water can move across the membrane, but because the concentration of solutes is the same inside and out the cell, there will be no net movement of water. Okay? In contrast, if we take a look at the middle panel, here we see a situation, again, a cell compared to its surrounding environment. Notice that the cell is hypertonic relative to the outside environment, which we would say would be hypotonic. Because the cell is hypertonic, has high concentration of solutes, again, water can move in both directions in and out, but the net movement of water will be into the cell toward that higher concentration of solutes. What will happen? Well, the cell might swell. It might even burst if we're talking about an animal cell, or if we're talking about a plant cell, the cell might just remain rigid due to the presence of the cell wall surrounding the cell membrane. Similar situation on the far left, only sort of the opposite. So here we have again a cell compared to its surrounding environment. Cell has a lower concentration of solutes, so we say it's hypotonic relative to its environment, which we say is hypertonic. Again, water can move both in and out, but the net movement of water in this case will be out of the cell. What will happen? Well, the cell will essentially shrink. Okay, so again, profoundly important concepts with lots of implications for living organisms. So finally, I just want to very briefly introduce some concepts that relate to movement of solutes across cell membranes. And what I want to point out is that there are a whole host of proteins, various transport proteins, that are found within the membranes of living cells. So just a few of these different types of transport proteins. First of all, if you focus on the middle panel, what this is illustrating is facilitated diffusion. So you see the phospholipid bilayer, that's your cell membrane. These green structures, those are various proteins that they facilitate or allow specific molecules, ions, to move across a cell membrane. So they just allow or facilitate diffusion of specific molecules across the cell membrane. Okay. Also, if you take a look at the far right panel, this is illustrating active transport, very similar to facilitated diffusion. But what I want to point out is that active transport can use energy. Energy typically in the form of ATP to actually pump specific ions or molecules from one side of a membrane to another. So what this has the implications of doing is actually forming a high concentration of solutes, again on one side of a membrane or another. And again, the point here is that active transport, of course, requires energy, typically in the form of ATP. And just as one example, later on we might talk about the nervous system of humans and how your nerve cells function. Well, our nerve cells function in large part because we have these active transport proteins embedded in the membranes of our neurons that actively transport sodium and potassium. And so the movement of those ions is what allows our nerves to function properly. And again, we'll discuss more about that at some later time. So that was very brief, but this concludes our brief introduction to lipids, membranes, and the movement of water